Okay, it's just a minute to the start time, so I think we can get ready to start with the next talk in this session. So we're at the session on analysis of complex systems, and this is the third talk in that session. The talk is on CABEAN 2.0, efficient and efficacious control of asynchronous Boolean networks, and it's a collaboration from colleagues at the University of Luxembourg. So Jun Pang is going to present, and I think you're ready to go. So 15 minutes talk and 5 minutes for questions. Over to you.
Okay, so yeah, let's start. So thank you for the introduction. So today I'm going to present to you our software. So actually the name of software is CABEAN, which can achieve very efficient and effective control of asynchronous Boolean networks. Here we use Boolean networks as a model for gene regulatory network in our cells. Okay, so so most of the work has been done by my students. I'm just here to present it. So first, let me to introduce what are the background, what are the motivation for our work. So actually, we are motivated by so-called cellular reprogramming. So here I have a picture here to draw so-called this kind of Wellington landscape. So here he viewed the development of the cells as like a ball rolling down to a very hilly landscape. So once the ball rolling down through the landscape, it gets more and more specified. And eventually, the cell will get into one of the valleys, and then that is corresponding to the cell types. Right. So what is behind the landscape actually is a functional network of many genes in our cells. So the main point is existence of these gene regulatory networks. So what do we want to do with the cell reprogramming? So if we have given like one type of cells, the reprogramming can somehow reprogram the cell into a different cell type. For instance, you have a ball like here. You see my pointer here. If you have ball like in this here, so that's represent one type of cell type. But the idea is you want to reprogram the cells in order to get into another valley, represent another type of cell type. And this has a lot of like applications in biomedicine. Okay. But if you want to perform this kind of like reprogramming, there's many different ways. For instance, you can do this like for following the arrow here, you can do this as completely de-differentiation. So you make the ball back to the initial point and let it to roll down again along this landscape, and hopefully it will end up into desired cell type. And another way is you can go back to some midpoint of the landscape and then ask it to develop into a different cell type. But what is more attractive is so-called direct cell reprogramming. So for instance, I show here. So you have a cell here. I directly perturb some of the genes or proteins in cells and make sure that the cell will change its cell type to the more desired cell type. So that is our biological motivation for this. And this kind of like cell reprogramming have seen a lot of clinical applications. So here I just show you a small table showing you that for different disease, you can somehow reprogram some kind of the cells in our bodies and into these more designed cells, which is quite useful to give some kind of treatment for all kinds of disease here. So this has been identified and somehow approved to be feasible. Okay, so as I said, what is behind this landscape is so-called gene regulatory network. So it is a function network showing that how the genes interact with each other and so on. There's different ways to achieve to achieve this kind of cell reprogramming. This is more like traditional method in the biology. What they do is they try different ways to perturb the genes in the network, and often this is supposed to be quite time-consuming and very expensive to do it. Right. So for us, it's more interesting to do it is on the computational model. So we want to have a model of the underlying gene regulatory network, and have based on that, we understand this functioning biological process of this network, and from there we want to identify which combinations of genes, if you apply perturbations, that will allow you to achieve your reprogramming. So the purpose how to change one cell type into another. right? Of course, if you talk about mathematical models of this kind of gene regulatory network, there's plenty of different type of models. And here we focus on the so-called Boolean network. Okay, so for Boolean network, this is a very high level like abstractions of the underlying gene regulatory network. So the network just consists of a set of Boolean variables, and each present like one genes in the network, and the state of these variables can be either 0 or 1, just showing that whether indicate whether the gene is active or not. So you have this, why this is called a Boolean network. And for each of the Boolean variables, there is also assigned a so-called Boolean functions describe how the value of the variables can be changed, and how. So here, if you look at the picture here, I show some of the Boolean function that in the picture. So it shows that how the other genes can regulate these genes in order to change its state. Of course, if you look at the dependency of the variables, then you can draw the network here. So the network will show that how the genes depends on on each other, right? And then the values can be changed according to the Boolean functions and so on. So if you have the set of Boolean variables and set of Boolean functions, then you can look at the transition system or state of the system, right? Okay, if you look at the semantics, there are actually like two ways you can update the values of the Boolean variables. The first one is called asynchronous. So every time you update all the nodes in the network all at the same time. So this is called the synchronous Boolean network. Somehow this is considered not so realistic because in our cells, the different reactions take a different speed. So it's more realistic to consider so-called asynchronous Boolean network, where at each time point, you just randomly pick one of the nodes, the Boolean variables, and update its value according to the Boolean functions. Of course, it depends on the values of the other Boolean variables and so on. But at each time, you only update one of the variables. So this is called asynchronous Boolean network. Then, based on this, you can actually draw the transition system of the Boolean network. Of course, if you have n variables, Boolean variables, the number of states in the system is huge. It's 2 to the power of n, right? So you immediately, if you want to analyze this kind of network, you immediately have the problem of state space explosion, right? And as what I said at the beginning, what is more interesting is if we have this kind of transition system, it's very interesting to look at what are the attractors in the transition system. So if you look at the figure I draw here, I only have a small network of 3 variables, and here I give the Boolean functions, and I can draw the transition system for this particular Boolean network. And if you look at the picture here, there are actually 3 so-called attractors of this transition system. So if you starting from any point, if you run the system long enough, eventually you get into one of the attractors. Actually, the attractors has its meanings. The attractors often corresponding to the cell types, as what I discussed before. So attractors corresponding to the cell type. Right? So what is interesting is if you have Boolean network, it's very important to identify what are the attractors and so on. Okay, so if you have attractors of this kind of like dynamical system, then you can compute what are so-called strong Bayesian and weak Bayesian. Because you have attractors, the strong weak Bayesian simply covered all the state which has a path into the attractor. So here, if you look at the picture here, so I have attractor here depicted as this red node, and I picked all the nodes here that have some kind of path going into this attractor. From like 1, 0, 1, you can go through 0, 0, 1 and go to 0, 0, 0. Okay, so the weak Bayesian simply covered all the state which is possible to reach the attractor. Okay, and then I also can define what is so-called strong Bayesian. Strong Bayesian covers all the state which can eventually end up into the attractor, but they cannot reach any other attractor. Okay, so here you see that all the paths from those states, they have to end up into the attractor of the attractor. For instance, if you look at the picture here, so I have the same attractor as the previous picture, but here the strong Bayesian only cover the attractor state and 0, 0, 1. But all the other states are not in the strong Bayesian because they can go outside, they have a path go into a different attractor. Right? So for instance, if you look at this one, this have a path go into another attractor. right? But here for strong biasing, I require all the paths from the set had to eventually get into the attractor. So that's the difference between strong biasing and weak biasing. But actually, that will be basic for us to derive all the algorithms for the control of a synchronous Boolean network. Okay, as I said, it's very important to have efficient algorithms to compute what are the attractors of given Boolean network. And here, actually, all our algorithms are actually built on the idea how to have the divide and conquer approach to compute this attractor, because it's very expensive if you take the network as a whole and compute all the transition system, then you want to find out what are other attractors. So we have to have a smart way to do this. So that's the work we published a few years ago, that we can do this decomposition-based method to perform these attractor detections. Here, just show you that. So here are the network, like real life network we identify in the literature. You see that they have a different number of nodes, starting from like 30, and some of them, we have a very large network of like 100, almost 200, 300. So if you think about the size of the transition system, you can think about 2 to the power 300. So that's huge, right? You cannot directly build the transition system and analyze the attractor. So that's why the so-called decomposition method is really important, apply an important role here. So you see here, even for a very large network, if we apply so-called decomposition method, so you read this kind of columns, the numbers in red colors, you see that we can compute all the attractors, the number of attractors very fast. Just, I don't know, here, like, I don't know, 7,000 seconds, you can do that. So around like 1 hour, deal with this such a large network, you can compute that, no problem. Okay. So then we are talking about the so-called control of Boolean network. So at the beginning, I talked about cellular reprogramming, but if you the reprogramming is recast as a control of Boolean networks. So here, so basically, there are 2 types of controls we can formulate, we can define. One is so-called source target control. What that means is you have like a given source attractors, you can somehow make a perturbation to the network and then let the network to get into so-called target attractor. So that's what I said at the beginning. If I have one cell type, if I want to reprogram it into a more desired cell type, right? And second control is called target control. So this is different from first one, because first one we are given what is our starting point. We have the source attractor, but here, we are talking about you want to program the cells into a target attractor, no matter what are the initial state of your system, of your network. From any state, if I perturb the network, you will go into the target attractor. So these are the 2 types of control problem we have defined. Of course, if you want to have a control to the network, you can also apply this control at one time. So you identify all the perturbations, then you apply it, and then that's it, right? So this is one step control. Then you can also do this in a sequential step. So you want to say, I want to apply my control at one time point, and then let the network to evolve at some point later, and then apply another set of control, and then I will see what happens. I want to drive the network into the target attractor. So you can have the one-step control or sequential control. And of course, if you look at what kind of perturbations you can apply to the network, we also define 3 types of perturbations. First is so-called instantaneous. So you apply the control, then you release it. So this is called instantaneous control. And second time is temporary control. So you apply your control there and hold down the control for some time, and then you release it. And in the end, you have the permanent control. Simply means that you apply the control permanently. Simply means that you set the value of the node as 1, and it's fixed forever. But for the above 2 types of controls, then you set the value, then you release it. So this is 3 types of control you can apply to the network. So if you bring everything together, so this is this number of control methods that we can derive, right? So you have the source target control, and depends on whether you apply the control at one step or a number of steps. Then you have this depends on what kind of perturbations you have. You have the 6 number of control method, and for target control, because of the type of perturbation you want to apply, then we have 3 types of control here. So this is all the control method we have implemented in the software, and that's it. So I think I'm a little bit out of time, but I'll give you a very quick idea how we actually achieve this. Okay, so for the one-step control, actually we rely on so-called strong basing. So if you want to go to in the target attractor, I take the target attractor, compute what is strong basing, then you compare your source attractor to the strong basing, then to find out which are the minimal perturbations that you can apply, and you apply it, because then you from that source attractor you jump to the strong basing. Of course, by the definition of the strong basing, eventually it will get into the attractor. Okay, so this is one step in instantaneous control. Of course, if you look at the temporary control and permanent control, then this is more involved. Instead of look at the strong basing, what you can do is you can look at the weak basing. So what you need to do is once you apply the control, you want to make sure that at the moment you release your control, the system, the network gets into state of the strong basing, because then you're guaranteed to end up into the strong basing. So that's the one condition you have to check. And second condition is that all the state after apply the control, that must be in the so-called strong basing of the strong basing. So you have the attractor which has the strong basing, but once you apply the control, then all the state there must be in the strong basing of that particular state. So that's guaranteed that you eventually will get into the attractor. So the idea is for temporary controls, they use a weak basing instead of a strong basing. Okay. So here give you a very quick result here. So this is number network we have show you before. Remember that here you have this number of like control, you only apply them one step. You see that this is instantaneous control, and the second line is temporary control, and the third column is the permanent control. You see here, if you have a temporary control, the number of perturbations you have to apply is much reduced. Right? So you see that from here is 9 or 20, and if you use temporary control, only is 3 and 4. And temporary control is probably the more designable the method you want to apply. And of course, if you apply your control at like a sequence of steps, you can further reduce the number of perturbations you have to apply. So for instance, here you see that here, if you do this in a sequential way, the number of perturbations you have to apply only like 2 perturbations. This is like reduced one by one. But this is a source target control, and here gives the computation time. Different method, and if you want to compute the solution, how much time you have. And here you see that for large network of 300 nodes, we can still do it within like 1 hour. Okay, so I think more or less I'm there. So I also talk about the target control, programming, reprogramming from any initial state. What we need to do is we look at weak basing and identify so-called schema, and every schema give a potential control, but that does not give you the minimum number of perturbations you have to apply. But so that for every schema, what we do is we enumerate all the possible combinations, and then we check everyone. So that makes our targeted control a little bit more expensive. In general, it also works quite well. But here, the difference between target control and what we said before, source target control is that we are not giving a specific initial state. So from any initial state, if you perform this kind of perturbations, then you are guaranteed to get into the target attractor. So here again shows what are the results. So this shows that how effective of the target control by looking at how many number of perturbations you have to apply here. So you see here the number of perturbations are really small. Even you have a large network like 300 nodes, you only need to perturb like one or 3 nodes, you can get into the desired attractor. So I think that's more or less I want to say here. So the main message I want to tell you is that, so I have the software allows you to control large asynchronous Boolean networks. You have different perturbations, but often the temporary perturbations is preferable. And the sequential control can further reduce the number of perturbations. And we also have like validations of our identified perturbations, which is in several networks, the identified nodes are really consistent with the existing biological knowledge. Okay, so I think I have to stop here. So thank you for your time. If there are any questions.
Thank you, Jun Pang. Okay, do we have any questions? What do you see as the next stage of this work? For future work, what are your
okay? Yeah. So for me, there's several ways to continue. First, here probably I didn't make it very clear. The perturbations I'm talking about is at the node level, because I have a node. I want to change the value of the node to fix to some value. This can be done in biology. But nowadays they have shown that instead of control the node, it can also more desirable to control the edges, right? Because we said the genes are connected by edges, but you can also try to control the edges. In some time, this is more preferable because it has less side effect to the network. Okay. So for me, the natural step is to see whether we can lift other computational methods for this kind of edge perturbations. Of course, another one, I want to go even further to scale up this method, because we know that in our body, within the cells, the size of the gene network is, I think, sometimes it's 500 or even 1,000, several thousand. So I want to see how can I go into that scale of networks.
Okay, sounds interesting, and it's good to hear the passion in your voice when you talk about it as well. So we look forward to hearing more about it. Okay, thank you very much. Thank you for that.